everyone. In the series in biotechnology, today we talk about the gene editing, where the modifications or alterations are made in the genome or the gene basically of our interest that can be utilized for various biotechnological purposes. For the genome or gene editing, various tools are utilized that basically causes these modifications to take place. The primary gene editing tools involve nucleases, as these nucleases create double-stranded breaks in the DNA that can then be endogenously repaired by the homology-directed repeat or non-homologous end joining, the two mechanisms we discussed in our previous session, and how and what are the kind of outcomes that these mechanisms result into. So, talking about the gene editing tools or the major nucleases, the earlier used were what were called as the homing endonucleases. Then there are zinc finger proteins that contain the zinc finger DNA binding domains and the nuclease part, which is basically the FOX1 endonucleases. We discuss about these zinc finger DNA binding proteins. Other are what are called as the talons. So, these talons are again the nucleases that have this FOX1 part, but then they have got these tail-like and nucleases attached to the tail-like DNA binding domain that give more specificity as compared to ZFN, and we will discuss in detail about that. And the most recent that you must have heard about is the CRISPR-Cas9 system, where the Cas9 protein associates with the CRISPR DNA molecule. Basically, it is an RNA-guided DNA repair method. It associates with this CRISPR RNA that is complementary or basically identifies the homologous regions in the DNA that needs to be edited, and there are specific sites, and then it repairs and basically create the double-stranded breaks that can then be repaired. So, this is the most efficient and very widely used method nowadays. So, we discuss in detail, but before going to that, let's look at the specific features of these kind of nucleases. For the zinc finger protein nucleases, as you can see, the recognition site is a zinc finger protein. The nuclease site is the FOX1 nuclease that is utilized. The target sequences are typically these kind of sequences which are there, and it is around 18 to 36 base pairs in nature. The specificity is provided by this DNA binding domain, and this can be a small number of nucleotides can be there, but it can recognize a little bit of mismatches as well. The targeting limitation, there are certain limitations to these method as well. I have just precisely summarized it in this table, and we'll elaborately talk about these nucleases subsequently. So, the targeting limitation is basically that it is difficult to target particularly the G-rich sites. The difficulty of engineering, because creating the diversity in these nucleases is again a cost and time burdening process. Then the difficulty of delivering is as well there, because these nucleases need to be delivered, and introducing them into the viral vectors to introduce into the cells to be targeted, the DNA of the cells which needs to be targeted, it is again a challenging task. However, still ZFN forms a very important tool for causing the gene editing. The talons, as I said, they are basically transcription related these effector nucleases. They also, the recognition site is basically these talon sites, which we will discuss in detail. It again contains FOX1 nucleases. They typically recognize these 14 to 30 base pairs, and there is a specific base pairs at 2 points which are recognized by these talon binding domains. The specificity is provided, and only a small number of potential mismatches are allowed. The targeting limitation is again that the targeting the talon monomer is basically a little problematic here. It's a little challenging here, particularly when there is recognition of T nucleotide. Again, difficulty in engineering, less as compared to ZFNs, but still for complex molecular cloning methods it requires that. And there is difficulty in delivery, again less than what is there in the ZFN, but because of the large size of these functional components, it becomes a little difficult to introduce them into the cells. So these are some features of the transcription activator like effector nucleases or the talents that we talk about. This is the CRISPR-Cas9 system features. So CRISPR stands for basically cluster regulated transcriptional repeats which are there. We will again discuss in detail about them. They have the recognition site which is basically a single strand RNA. We will see this is an RNA guided DNA repair system. So what we require, the recognizing site is basically a single strand guide RNA, and then the nucleases is basically provided by this Cas9 protein which is a nuclease. The target sequences are certain sequences which are the PAM sequences, the small PAM site sequences. The specificity is it is highly specific and it is basically very less mismatches are basically recognized, but it is a very highly specific system. The targeting limitation includes basically the strictness of this PAM sequences. Not much versatility is obtained in that, so it creates a kind of a specificity, targeting limitation. Targeting engineering, it requires the synthesis of this guide RNA as well, the oligosynthesis, the cloning and etc., but these are all now standardized and therefore modified to create more efficient process. As far as the delivery is concerned, the most common is the Cas9, as I said, protein delivery method, and it is quite an efficient method of delivery and does not create much difficulty, but still methods are improved upon so that the entire system can become more efficient. If we look at the timeline, in the 1970s the homology recombination was the only method for genome editing that was utilized. But subsequently in 1980s and in 1990s these ZFNs were discovered and they were used to target the particular sequences. As you can see, then the versatility was also created in ZFNs, and further on in 2000 and 2011 basically the talents came into being and then the talents were utilized, and then early in 2012 the CRISPR sequences came in and this CRISPR system was basically developed and many improvements have been there for CRISPR method, and we all know that in 2020 the Nobel Prize in Chemistry was given for the discovery of this CRISPR-Cas9 system, which is a highly efficient system for genome editing. Talking about now the genome editing nucleases in detail, first we deal with the meganucleases which are called as the homing endonucleases as well. They are the members of this LAGLI-DADG. This is the conserved amino acid motif that interacts with the DNA family of endonucleases. So initially when they found out that the double-stranded breaks stimulate the efficiency of the homologous recombination, then these meganucleases were utilized to increase the efficiency and cause the genome editing, and they belong to and they recognize this amino acid, basically conserved amino acid motif that interacts with the DNA. The naturally occurring enzymes that recognize and cleave the long DNA sequences which were around 14 to 40 base pairs. So they recognize these sequences, these amino acid motifs, they recognize these sequences and cause the double-stranded breaks which are basically there. They make extensive sequence specific contacts with their DNA substrate and therefore show exquisite specificity. However, the binding and cleavage domains are not modular. It's the same protein, so different module proteins are not involved in the binding and as the cleavage. As we will see in the subsequent nucleases, we have separate domains for the binding and the cleavage part, and because of this overlap in the form and function, they make their repurposing challenging and they limits their utility. So we cannot utilize these meganucleases for widespread genome editing, because these binding and cleavage domains, when they are modular or separate, we can use diverse binding domains and diverse cleavage domains in connection with each other, and therefore they provide repurposing as well as versatility for the nucleases to cause different kind of things to occur. So this binding and cleavage domains are not modular here. That gives a limitation to these homing endonucleases or the meganucleases. Looking at the zinc finger nucleases discovered by Chandrasekharan, Srinivasan Chandrasekharan, these are basically the fusions between a custom designed cysteine histine dimer zinc finger protein and the cleavage domain of the FOX1 restriction endonucleases. So basically the cysteine, dicysteine and dihistidine zinc finger proteins are there. These are the DNA binding domains, and these fusions, as you can see, so at least 3, we will see at least 3 these molecules are attached here, zinc finger molecules are attached here to give rise to the zinc finger binding domains, and they are attached basically, and they contain different kind of residues that are joined together, and recognition sites which are basically there, and they associate with the FOX1 endonucleases which are there. An individual cysteine histidine diamine dimer zinc finger consists of approximately 30 amino acids, which include 2 anti-parallel beta sheets opposing an alpha helix. Now these are the family of DNA binding proteins which are very important, and I hope you understand this terminology, which is basically discussed in terms of the protein structure. So these 30 amino acids are arranged in 2 anti-parallel beta sheets that oppose an alpha helix. Now these zinc finger proteins or nucleases, they recognize the target DNA in a modular fashion. Each protein consists of at least 3 zinc finger domains. Each domain through its alpha helix residues interacts with a 3 base pair sequence within the major groove of DNA. So these are the 3 base pair sequences that are basically recognized, or when they arrange as a dimer, so they recognize 2 sets of 3 base pair sequences as shown here. So they recognize these 3 base pair sequences, the DNA zinc finger binding domains, and they cause this specificity binding, and then these nucleases cause the double stranded breaks to occur. So these 3 base pair sequences and how they interact with these sequences, these proteins interact by their alpha helices, which gets into the major groove of the DNA. So we understand the structure of the DNA, and we understand the structure of the proteins, and this tells us how these proteins and DNA interacts or binds together for causing different kinds of functions. So here we talk about the zinc finger nucleases. The FOX1, the FOC1 basically the FOC1 type 2 restriction endonuclease forms the domain that cleaves the DNA, which can be adopted as a dimer to directly target sequences within the genome for effective gene editing. So this FOC1 type 2 restriction endonucleases that cleaves the DNA. So the DNA binding domain, zinc finger proteins, gives the binding, the specificity part, and they basically cause the DNA to be cleaved. After the DNA cleavage by ZFN, the DSBs at a specific locus of the genome is initiated, creating the desired alterations in subsequent endogenous NHEG, NHEJ or HDR repair. So these mechanisms we have already discussed in detail. So how these double strand breaks are created by zinc finger proteins we have discussed just now. The target sequence recognition and specificity of ZFNs are determined by 3 major factors. The first is the amino acid sequence of each finger. So that 30 amino acid sequence is important and varies and can vary from one finger to another, but the recognition and the specificity of the target sequence is highly dependent upon that 30 amino acid sequences. Then how many number of fingers are involved? As I said, at least 3 fingers are required for the nuclease to work, but again the specificity and the recognition will depend upon how many number of fingers are there. And finally, the interaction of the nuclease domain. So this is related to the zinc finger part, the DNA binding domain, and then how the nuclease domain is interacting. All these factors, 3 factors, will determine the target sequence recognition and specificity of ZFNs. Now, what are the limitations and concerns? One major concern associated with the use of ZFNs for genome editing is the off-target mutations. And these off-target mutations can disrupt certain important genes or activate certain toxic genes, leading to the cytotoxicity. So this off-target mutation is again a very difficult aspect, because it is recognizing just 3 base pair sequence, which is providing the specificity. The second limitation or difficulty is basically associated with the construction of the zinc finger arrays. It remains challenging to create ZFD that can effectively recognize all DNA triplets, especially those of 5' CNN or 3' or 5' TNN 3' variety. So these T containing triplets or C containing triplets are difficult to be recognized by the zinc finger domains. Because it is recognizing the triplets, it is very difficult and challenging to engineer these ZFDs that can recognize all these kind of different triplets. So these are some of the limitations that is associated with ZFNs. Although these ZFNs were utilized and still utilized at a larger scale, but the advent of different genome editing tools such as talents, so in between, as we saw in the history of the timeline, of the discovery of genome editing tools, initially homologous recombination, the earliest method, then came ZFNs, then their limitations were somehow or somewhat countered by the talents, and then came the CRISPR-Cas9, which is today most efficient method of genome editing that has been utilized. So what are these tail nucleases? These tail nucleases are based on the transcription activator-like effector, acronymed as tail proteins, from Xanthomonas bacterial species that have a DNA binding domain and an effector domain. So this is the structure, architecture of a talon, and we will discuss in detail. So again, tail is basically the DNA binding domain and the effector domain, which then get associated with nucleases, primarily the FOC1 nucleases, and give rise to these talons which is there. So looking in detail on this tail architecture, the N-terminal domain harbors the bacterial secretion signal and a non-specific DNA binding activity that is required for the general affinity of the protein to DNA. So the first aspect is to bring the protein to the DNA, proximally to the DNA, and then basically the specificity is required. So this N-terminal domain basically causes this proximal binding activity that is required for the general affinity of the protein. It also contains a segregation signal that implies that the tail molecules can be secreted outside the cell as well, and again makes its purification easier. The C-terminal, if we look at the C-terminal, it harbors an interaction interface for the plant transcription factor 2A. Now these tail proteins were isolated from a bacterial species that was associated with plant. So it was a plant pathogen basically. So this plant bacteria, they contain a plant transcription factor 2A interface, interaction interface, 2 functional nuclear localization signals, and an acidic activation domain. So this is an activation domain. This is then when the protein is formed, it is localized into the nucleus. or it can be also secreted out of the cell from the nucleus, then it goes to the endoplasmic reticulum, and it follows that secretory pathway, and therefore it is these proteins are there. The key part is this, repeat. The key to this specific and programmable DNA binding is the central repeat region, which consists of a variable number of typically 33 to 35 amino acid long tandem repeats. So these tandem repeats are very important, and particularly the amino acids at this position 12 to 13, which is termed as the RVD, or the repeat variable diresidues. Now these repeat variable diresidues give rise to the engineering of variety of talents that can be there. Again, here certain RVDs are given and certain sequence specificity where these RVDs can bind. So these repeat variable residues are the one which initially interacts with the DNA sequences, because this is a DNA binding. So this is the key DNA binding domain, and in these tandem repeats, the 12th and 13th position amino acids, the residues are very important and causes specific binding to various kind of DNA molecules. So this contain the variability and the variety or the diverse nature of talents that can be created. Two amino acids that define the DNA specificity of a tail are located at position 12 and 13 of the each repeat and termed as the repeat variable diresidues. DNA binding specificity is determined by RVDs with non-specifically binding to C nucleotides. These are the two amino acids, the ND specifically binding to C nucleotides, the HN to A or G nucleotides, NH to G nucleotides, and NP to all nucleotides. So these are diamino acids, and their specificity to different nucleotides where they can be bound, which is also given on the side here. So this dinucleotide within this repeat region is very, very important for the specificity of the molecule. So general affinity can occur because of these domains, a recognition can occur because of these repeats, but the specific binding is because of these 12 and 13 amino acid, and based upon what variable residues are present here, they can bind to different specific nucleotides out here. So these nucleotides, which are basically present at the double-stranded breaks, recruit these, which is basically associated with the bounding at the DNA, and then the nuclease can create a double-stranded break which is there. So these are the different diamino acids which are specifically binding to certain nucleotides on the DNA. There is a one-to-one correspondence between the RVDs and contiguous nucleotides in the target site, constituting a strikingly simple tail DNA recognition cipher. So this is how basically the tails basically attach to this, to the DNA. Now tails form a right-handed superhelical structure that wraps around the DNA. Each repeat forms two alpha helices that are connected by a loop region in which the RVDs are placed. So this is the crystal structure has given this structural organization. So this was basically the sequential organization, and structurally these tail proteins, they form the right-handed superhelical structure that wraps around the DNA. They're placing the two nucleotides onto the nucleotides that it binds to, and these repeats are basically connected by a loop where the RVDs are present. Again, the functional endonucleases, FOC1, is factitiously fused to DNA binding domains to create site-specific DSBs and thereby stimulate DNA recombination to achieve the TALEN-induced targeted genomic modification. Let's look at how that happens. Now, a TALEN module is designed in pairs to bind the opposite DNA target loci with proper spacing of 12 to 30 base pair between the two binding sites. So again, you can see that two molecules of talens are attaching. This is our target site, and this is basically the area where the editing will occur. So this editing area is again recognized by, so these are the nucleases, this is the DNA binding domain, and these are the nucleases that recognize with a proper spacing in between, and then they can make double-stranded breaks here that will again be repaired by the HDR or the NHAG method. So this is all about the talents, how they work. The talents offer two distinct advantages. First, no selection or directed evolution is necessary to engineer tail arrays, dramatically reducing the amount of time and experience needed to assemble a functional nuclease. So there is no, just as we saw, there is no selection or directed evolution is necessary. The tail arrays can be dramatically, more easier way they can be assembled and, to assemble this kind of nucleases which is there. Talents have been reported to show improved specificity and reduced toxicity when compared to some ZFNs, potentially because of their increased affinity for target DNA or greater energetic penalty for associating with base mismatches. Lastly, there are certain limitations associated with talents. The talents are substantially larger than ZFNs. We saw that they contain 30, 45, 35 nucleotides repeats and have a highly repetitive structure, making their efficient delivery into the cells through the use of lentivirus or a single adeno-associated virus particles challenging. And then primarily technical hurdle for cloning repeat tail arrays is the design of a large scale of identical repeat sequences. So in designing these identical repeat sequences create a technical hurdle, and again delivery is challenging, but talents are better than the ZFNs in terms of their manufacture, their creation, their engineering, as well as the versatility that they can create. And therefore, they became the choice of genome editing tools in the past above and over the ZFNs. So, we discuss about the CRISPR-Cas9 system in detail in our next session on the genome editing tools. So, that's all for today. Thank you.